good afternoon. Now moving from feed efficiency to resistance to gastrointestinal parasites. And all this delay was, now we have Gabriel Chapesoni connected. And he's the expert in genetic resistance to internal parasites. And the idea, I'm going to give a short, quick background and explain the exercise that we have. Following on the presentation by Rachel today, the gastrointestinal nematodes gene is one of the main constraints for sheep production. And what we know is that lambs are the animals that are most susceptible. But there is a time in the ewe life, that is around lambing, that when they experience this phenomenon called periparturient rise, that for me now on is going to be PPR because I cannot pronounce the long name. And what happened in the cycle of the parasites is that basically what we have in the abomasum and the rectum, we have the adult worms, and the eggs are liberated in the feces, which develop and go across these two phases, L1 and L2 of larvae, that they stay in the feces and eat based on the feces, and the L3 that move from the feces to the grass and is again eaten by the animals. And this is the cycle of the worm. And usually what happens is that the stage from here, that L3 that transforms to adult, or also called L4, and produce the eggs, is like between 2 weeks, between 2 and 3 weeks. FEC, or fecal worm egg count, is the number of worm eggs in the feces. So it's the sample that is collected here, the one that we analyze. And the number of eggs is an indication of the number of adult worms that is in the guts of the animal. And it's basically the most, even it's complex to measure. And FAMACHA, for example, that Rachel mentioned today, is much easier to implement, is still being the most widely used method. FAMACHA, that is more or less the same picture that Rachel have today, and it indicates is the measurements in the mucosa around the eye to compare the level of anemia, being pink and white, clear symptoms of problems of anemia, and it's a clinical identification of the anemia that might be related with other reasons, but in case that is being due to infections of parasites, is basically due to one species, that is the Aemonchus, because Aemonchus produce anemia in the animals. The applicability of this is usually in normal situations in a farm. Well, what you do is sampling the feces from a group of animals, so you can identify the moment that you have to use anthelmintic for treatment of the group of animals. So it's basically a sort of strategic use of chemicals for treatment of parasites. In the case of FAMACHA, because it's easy to use individually, what you can define is the individual treatment of animals. So it's not just a strategy to reduce the use of chemical to reduce the impact of the resistance to anthelmintics in this treatment for the groups of animals, but it's also for animal by animal. Both are also potentially being used for genetic improvement to increase resistance. FEC is the one that is basically being used in most of the genetic evaluations. Based on these two LAMPs, we have two examples, two analyses. One is FEC data on corridor LAMPs with the idea of checking the structure of data. It's a subsample for genetic evaluation. We don't have all the data. We have only the phenotypes and fixed effects, just for you to consider the potential fixed effects in the model, and the normalization effect. And then in the second analysis, or second database, is for the PPR in use of different breeds. And in this case, it's experimental data. It's actual real experimental data. And the idea is for you to define the model and to compare the results between the breeds or among the breeds. In the case of the example with the gene infections, I need to give you some background just to help you understanding the database. Genetic selection for FEC is based on the estimating breeding values. Individual FEC is recorded in the LAMPs after weaning. The fecal samples are collected directly from the rectum of the animals. After the identification of the animal, you have the sample, you have to identify with an identification of the animal, sent to the lab, and then FEC is counted to establish or computed in a lab. In Uruguay and in other places, the fecal samples, well, in the genetic evaluation, basically is two measurements of FEC are used. No? So you have lambing is around September, then weaning is 3 months later. You have the first drench to ensure that all animals are in the same conditions, so they are clean. Oops. Yeah. And now, okay, go out. How can I? Okay. And then you have first range. You have to check after range that FEC is actually 0 to be sure that the animals are clean. And then for each contemporary group, they select a group of, I don't know, 15, there's in the protocol between 10 and between 15 and 20 animals. And you monitor the level of FECs in the group to see the evolution. When FEC is higher than 500 and there's less than 20% of the animals of that sample with FEC 0, you have is the moment for the first sampling of all the animals in the contemporary group. And this is FEC 1. Alternately, and because it implies to increase the accuracy of the measurement of FEC, of resistance to the parasites, the suggestion is to do 2 measurements. So after doing FEC 1, collecting the sample, sending to the lab, again, a second drench is applied in the animals. You wait and check that effectively the animals are clean. FEC is 0. You wait again, monitor the animals, and when you reach the same parameters, then you have the second sampling. So what you're going to see in the data, in the Excel file that we send, is the animal identification, the year of recording that goes from 2008 to 2021, from 4 flocks, sex of the lambs, and you have 3 categories, male, females, and castrated, and the type of birth, single, twins, triplets, and quadruplets, identified with those numbers. And this is mandatory information for the genetic evaluation that includes other traits. And you have here body weight at the time of FEC1, body condition score, also recording the same animals in FEC1, and FEC1 and FEC2 as the traits to be analyzed. Gabriel, ¿quieres agregar?
No, I'm saying, only one comment. We usually have a body weight at this age when it's the similar age like the fecal count, but the special body weight when the fecal count was recorded was a special project. But usually the cordial breeder have a body weight in this 8-9 months. That is usually the fecal count date.
Okay, so there's an overlapping between this body weight with another weight that is mandatory within the evaluation. Different from what was done in the before, what you're gonna have an Excel file that says analysis 1 or something like that, very easy to understand. Then a copy with the scripts in SAS and the scripts in R to be analyzed, and the output of the results that we are expecting you to see. Basically, based on what is written in those scripts, you can try different things and you can do all different combinations. The idea is the basic, that is the beginning, that is uploading the database and checking that the database is being read properly. So you have to see the traits that I mentioned before, and the number of records is 3,964. Then you have the description, the analysis of the data, the generations of new traits, of new variables based on what you have, the GLM or general linear model to analyze FEC1 and FEC2, and the normality test for FEC. When I say generation of new variables, you will see this is description for the traits is, well, when you're gonna see in terms of flock year, the distributions, the number of animals for type of birth. Also to see if you need to put together different classes depending on the number. You think that at least you have to create the component, to put together the traits that will define the contemporary group for a genetic evaluation. Then analyzing type of birth to see what you can do. Then test different transformation of FEC, because you're going to see in the distribution that is very far from being normal. And see and test normality for these traits. And again, one important thing here is, well, this is the analysis to see how it works, the different combinations of this contemporary group defined as year, flock, sex, and type of birth. You can try to see if you can put together a type of birth within the other factors and analyze the effect of type of birth on FEC. And one important thing is to create the residuals and export the residuals to be analyzed here for the normality test, because you're going to see the different changes in the distribution of FEC based on the different mathematical transformation and based on the residuals. Gabriel?
Oh, it's okay. Only that we have there only an example of the normalization that usually use the logarithms, the natural logarithms, but in other countries they have cubic root or other logarithm in base 10 and base 2. You can try different transformation.
Well, and then for the second, in the second is about the gene infection induced. What it's observed is a rise in FEC in the use after lambing or around lambing. That is basically due to a reduction, a loss of immunity because of the stress around that season. And the problem is that the PPR leads to an elimination of worms, eggs into the pasture that could be an additional contaminations for the newborn lambs. What in this case is the experimental data collected on the experimental station where we are located, INIA-Las Brujas. And it's the comparison of 3 breeds here. That is, the first one is Creole sheep, is Frisian and Texel. So you have, in this case, rather than the lamb, here the information is about the ewe. So you have the age of the ewe, it's adult or hogged, the breed, the identification, the number of lambs born, lamb date, and we have here date 1, body weight, body condition score, FAMACHA and FEC for date 1. And then you're gonna see the same data with 2 in each of the things for 5. In total, you're gonna have 5 sampling dates that where also body weight and body condition score were recorded. So you can see, and part of the exercise is also to check in the data the different distribution and the difference between the lambing date and what you see in the different rates. In this case, it's not just the problem of the, again, and you will see the same information. You have a document with the scripts, and transforming the new traits in days in milk, lambs to see the distribution, the transformation of FEC, and analyzing the means for total sort of description of the database. Check for the fixed effects, lambs versus age and breed. A simple GLM to analyze the difference between breeds. You can even want to transform the database and do something continuous or with multiple traits. The normality again. And you can check alternative models. That means based on this model, you can adjust FEC by body weight, considering also the fact that the number FEC is the number of eggs, is the density of eggs in the feces. That means number of eggs per gram of feces. Depending on the size of the animal, the feces are different size. That means that the contamination that is being released is different also depending on that. And you can do it in terms, rather than eggs, in terms of the number of females, that is the number of eggs adjusted by gram of feces, and adjusted by the biotic potential of Haemonchus contortus, that is the species of the parasites that is more common in Uruguay. And then you also have to check based on this same model and explore how is the evolution around lambing for the 3 breeds based on FAMACHA, body condition score, and body weight. And you will see that there are different patterns.
I have a question.
Yes.
So when you're saying that some animals, depending on how much feces they produce, and on the kind of the kilogram of feces times the fecal egg count, produces kind of the how much this animal will contaminate the pasture.
Mm-hmm.
So did you calculate this?
Yes, yeah. And then there's a. It's not a very common phenomenon analyzed, but there are also Gabriel working in a few years ago in terms of the different evolutions and the abilities of those traits and what happens if you use different transformation and what is the contamination of that in the, because now the U by itself, as an adult, the immunity is higher in the adult. But the problem is that this effect has a contamination has the contamination effect on in the pasture for the lambs that are born and for the other lambs that are around.
So I mean, on the herd perspective, you want to get rid of the animals that contaminate the entire pasture the most.
Yeah. From a genetic point of view, we wanted to characterize also this breed because there is a sort of expression that or expectation that Creole sheep should be more resistant to internal parasites. But I mean, in terms of the genetic selection for FEC in lambs is highly correlated with resistance in FEC in the use. So it's not a problem of selecting, say, well, we're going to have to select this because it's an independent trait or the other one. But it has different implications in terms of the management, and the objective here is to see difference between breeds. Rather than selecting within breed, that is what a breeding program does, is, well, see if you have different behaviors in different breeds. I'm going back to the slides, the first ones. I think that you have all the, yes?
The acronyms, PPR, I'm aware it's also used for a disease in ruminants, small ruminants, but still the petty ruminants. So I don't know if there will be a confusion with what you now use it for.
You say the acronym could be generate confusions.
Yeah,
Well, maybe yes.
PPR, are you aware of
the gene that the...
No, a disease in small ruminants.
No.
PPR.
No, but this, I know. 5 o'clock. I mean, you can stay and you can work on the data, and the data was given so you can work and do. I don't like to use the word play because but you can analyze in the different ways and try different models and and check everything that you wanted. That's why we provide the data. One of the first things, and I want to go to the first one with the FEC data on lambs, is the information with in terms of the real information. As you see, we mentioned that to increase the accuracy, it would be good to have 2 measurements of FEC. And actually, the model that is used in the genetic evaluations includes a repeated measurements model, no, to make use of this potential rather than FEC1 and FEC2. But you can also see that the number of animals with FEC2 is lower than the FEC1 because there are 2 things that happens with measurements like FEC. One is that it's very demanding. It's not just the cost in terms of the analysis, it's time consuming, and in terms of taking the samples, analyzing animal by animal, sending to the lab. And just doing that and twice is something that many times is not all the breeders used to do end up doing both measurements. And also is the fact that, and that's why, is that you need to have the animals to be able to quantify the resistance or the potential resistance of an animal. You need to expose the animal to the disease, and that's why that monitoring to have a minimum level of expression of the disease to be able to quantify the environment. There have to be a certain level of burden of the parasite, and that is something that is sometimes quite difficult. Nobody who raises animals and wants to let animals to be exposed to the diseases, even if it's for a good purpose like identifying resistance. Well, the other thing that you can see is that there's different balance of data according to different flocks along the years, and that is something that is real in data, in the data that you have in the genetic evaluations. The same with the fact that that FEC is not normally distributed. That is clear, no? And they have beyond we use and propose 2 here. There's other participants in the group who have been testing other transformations of FEC to transform to normal transformation of the trait. But if you see the residual of the model of FEC, of log FEC, is normal, and that is the one that is basically used and is the final trait that is relevant to be normally distributed at the time of the data analysis. Gabriel?
Okay. Only 2 or 3 comments for the last presentation. So, for all the things that you say, Elly, it's very important to close very closely with the vet from the country. It's not recommended to import a protocol from another country. You must to know the epidemiology of your flocks. For example, for us, the first fecal egg count is more prevalent than Moccus contortus, that this is more important parasite. And in the second one, we have more Trichostrampus. For this reason, now we are trying maybe to move to a new protocol with only one measure, only for Moccus contortus. That is, we lost some accuracy of the genetic evaluation, but we gain more flock that registered this trait because it's a very complex trait. So the other recommendation for our experience is that in Uruguay you have 2 selection objectives. The first one is to reduce to increase the resistance in lambs, and the second is to increase the resistance in the spring race. But we have only one criteria because we do an experiment to estimate the genetic correlation between the 2 traits. So it was very important for us because it's more complicated to do this in the spring race because you must to test the ewe with the lambs. So for this reason, it's very important to do some coprological tests, to work with the vets. For example, in the case of the Famacha record, Famacha was developed in South Africa, and South Africa have very different weather conditions than in Uruguay. They have a rainy season. There is no rainy season. All the year is rainy in Uruguay. You must know almost know in the last 4 months, but you need to know your weather condition, your epidemiological condition, also to have a new protocol. For example, in France, they use we're talking about the natural infestation, but in France they use the artificial infestation, and you can work in a special nucleus and to do it and to spread all the genetic improvement from a nucleus to the other commercial population. So each country must to develop their own protocol for all the traits, but it's more important for these traits because you have two genetics, the genetics of the animal and the genetic of the parasite, as it's more complicated the interaction. So I think it's all from my part.
Mm-hmm. Yeah, well, that's the, I think that is mentioned in the PowerPoint, but all the data that we have been recorded and is the data that we share with you is under natural infestation. So again, also the levels of the expression of the trait depends a lot on the weather conditions for that infestation to happen. No? So sometimes this calendar that was so perfect that drained Feb 1 and so many months, and well, the sequence is the same, but when the sequence happens, it might be different depending on the weather condition. For example, now that it's very dry in Uruguay, and it might have lower density of parasites. And basically, the idea is that. So you have real data in terms of fixed effects, real distribution of data, and well, the considerations that Gabriel mentioned in terms of consider the real situations that you have and the cycles of the parasites in your conditions to be able to really optimize the potential response to selection. Again, heredability is not the problem. The problem goes from different sources, and that's what you need to optimize. And parasites in sheep, in lamb, is a serious problem. Anyone working with lambs knows that it's not theory, it's real problem, and resistance to antibiotics, to chemical treatment is a real problem, and is also happening not just now in sheep but also in other species, and it's very frustrating when you run out of options to control infestations that really might kill the lambs. It's a real problem. And you can also experience at research level sometimes. Any questions? Right.
What happens to your protocol if the parasites are resistant to your drench and it won't work anymore? Like as you said in research.
Yes. When I mentioned in research, I was basically thinking about the training, the feed efficiency test, that you need to be sure that the animals are clean before starting and sometimes, well, but nowadays you have the protocols in terms of combining different, and it happens for sheep and it happens for cattle, the protocols for parasites, internal parasites in general, are a combination of different drugs. One is to also reduce the progress of resistance of the parasites to the chemicals, but also because to spread the possibility of doing the chemicals. But sometimes you need to do. That's why you drench and you check that effect is 0, and sometimes you have to drench again. Yeah, it's a real difficulty in terms of, and it's spreading. Yeah.